Thank you very much for participating in this first inaugural webinar of our new journal, Aging in Cancer Research and Therapy, and Treatment, rather, an open access journal, which is, I hope, going to be able to speak for this area of importance in cancer therapy and treatment, namely that's focusing on older people, where there are, even though there are many new journals out and about, including one on aging and cancer, still a relatively unexplored area for specialist journals, and we hope to make a move in that direction. So it's a great pleasure to introduce the journal and the first webinar of this series, we hope. I am Graham Pavelitz from the University of Tübingen, Professor of Experimental Immunology, co-editing the journal together with Professor Per Tor Straten from Copenhagen. And it's a great pleasure to introduce this first webinar. Thank you very much, Viktor Umansky, Professor in the German National Cancer Research Center in Heidelberg. And this webinar will be hosted by my old friend and colleague, Rafael Solana, Professor at IMIC in Cordoba in Spain. So thank you all for joining. Good morning, good afternoon to all the participants. And Rafael, please take it away. Thank you very much for joining.
Okay, thank you very much, Graham. Thank you to all the people, to all the participants. In this inaugural webinar, we are going to talk about immunosuppressive tumor microenvironment mediated by myeloid-derived suppressor cells and their targeting. This concept of myeloid-derived suppressor cell was proposed to term the myeloid cells that originally were found to be accumulated in some particular pathological condition, particularly in cancer. This is a heterogeneous population that arise from altered myelopoiesis and disrupted maturation of myeloid progenitors. And they expand in these tissues, peripheral tissues, where there is some sort of pathology. The fundamental feature of these cells is that they are immunosuppressive. They have immunosuppressive activity, and they have been defined to be expanded in several clinical conditions, in particular in cancer. To talk about this subject, we have Professor Viktor Umansky, the professor of oncology in the University of Heidelberg. He's a leading expert in tumor immunology and melanoma immunotherapy. He has been working on that subject for many years. I have the pleasure to meet him at least 30 or 40 years ago. And he leads the melanoma immunotherapy group, and he has a strong background in the role of this of different cells of the immune system, but in the last few years, in particular in these myeloid-derived suppressor cells, in chronic inflammation in the melanoma microenvironment, and how these myeloid-derived suppressor cells and other regulatory T cells can affect or can improve or can induce tumor immune evasion. Professor Umansky has published at least more than 200 articles in top journals, including journals of the Nature group, Nature Medicine, Nature Immunology, and also Journal of Clinical Investigation, Cancer Research, Journal for Immunotherapy of Cancer, and many other journals. And he has really made a strong advance in the knowledge of this immunosuppression in different tumors, in particular in the melanoma model that he has developed. And I don't have to say anything else. I can give the talk to the word to Viktor Umansky, that can start his talk whenever he wants.
So thank you very much, Rafael, for this very nice introduction. So I'm going now to load the presentation. So do you see the presentation? Yeah.
Yes, perfectly. Thank you.
Okay, thank you. So thank you very much again for this nice introduction, and also thank you, Graham, for inviting me for this first webinar presentation or meeting. So as Raphael told, so we will discuss today the role of myeloid-derived suppressor cells in the tumor microenvironment and the induction of immunosuppression. So normally the myeloid cells are generated and differentiated from hematopoietic cell progenitors through different ways, and at the end they go actually to the fully differentiated, fully mature cells, neutrophils, monocytes, macrophages, and dendritic cells. However, in the process of chronic inflammation or tumor development, which is typical also for chronic inflammatory conditions, typical for tumor development, so there is a continuous strong production of inflammatory factors listed here, which can partially block this normal differentiation, leading to the accumulation of the not fully matured, immature myeloid cells with phenotype or morphology of neutrophils called polymorphonuclear MDC, and monocytes called monocytic MDC. So they have particular markers, especially very good seen in mice. You see them here. Unfortunately, in human, we don't have GR1 markers, so therefore we have to use for human polymorphonuclear MDC, so CD15 or CD66B or LOX1 as a marker, and the negativity for CD14. And for monocytic MDCs, this is the positivity for CD14 and low or negative expression of HLA-DR. Now, these cells are generated in the bone marrow, and just to reach tumors, so they need to make a long way, and this is done with the help of different chemokines. And in the end, actually, the process of establishment and accumulation of myeloid-derived suppressor cells in the tumor-bearing mice or cancer patients, so has this process two steps. So one step is MDC generation and migration, where transcription factor starts to replace a very important role, and you see this variety of chemokines and growth factors which are involved in this process, and cytokines. And the second step, which is very important, happened in the tumor lesions, leading to the full activation of MDC. And then there are several factors also produced by T cells and NK cells, like interferon gamma or TNF alpha, are important for the activation of MDCs. As Raphael told, the population is very heterogeneous and has also variety of mechanism of induction of immunosuppression, inhibition of T cell activity and NK cell activity. So I will list only a couple of them. So first of all, they are involved also in the induction of other immunosuppressive cells like regulatory T cells, or they can differentiate also in the tumor microenvironment into immunosuppressive M2 macrophages. Then they can impair also the lymphocyte homing in the secondary lymphoid organs. They produce also a lot of free radicals, factors like nitric oxide or reactive oxygen species, leading to the reduction, for example, of the expression of TCR zeta chain on T cells, and leading to the inhibition of T cell proliferation, or even to T cell apoptosis and nitration of T cell receptors. They also involved in the metabolic depletion, because T cells especially are very much depending on essential amino acid like L-arginine or tryptophan, and high activity of such enzymes like arginase or IDO can lead to the depletion of these factors, and T cell again undergo the reduction of T cell zeta chain expression and T cell energy. Similar to tumor cells and some other cells, so MDC can upregulate PD-L1 expression, which is a very important mechanism of induction of inhibition of T cell activity through the interaction with PD-1 on activated T cells. Also, one should mention the involvement of MDC in the production of immunosuppressive adenosine through the increased activity of ectonucleotidase CD39 and CD73, leading to the degradation of ATP to adenosine, which interact with adenosine receptor, A2 receptor on T cells, leading to the activation of adenylate cyclase and production of cyclic AMP, which in turn block the proliferation and activity of T cells. Now, we are working in our group with the preclinical mouse model characterized by overexpression of receptor tyrosine kinase in melanin-containing cells, leading to activation of MAP kinase, metalloproteinase, and spontaneous melanoma development very quickly in the age. And you see this here, the spontaneously developed tumors, and this is the lymph node metastasis. Now we found in these mice that there is a constant and gradual accumulation of myeloid-derived suppressor cells in the tumor microenvironment, which we can measure by expression of double expression CD11-BJ1. These cells in the tumors can produce some immunosuppressive factors like nitric oxide or increased activity of arginase 1. But the most important, what was told already, the peculiarity of these cells, it's a strong immunosuppressive activity. So it's necessary to show that they are really immunosuppressive, which people are using normally the inhibition of T cell proliferation, and T cell could be activated either antigen non-specifically or using, if we have T cell specific for some antigens, so you can use the peptide activation. And actually, you see that there is the strong inhibition of T cell proliferation after co-culture with MDC isolated from the tumors. Now we told about two subsets of MDC, polymorphonuclear and monocytic, and we studied systematically also the relative distribution of these subsets in different organs, so tumor-bearing mice, and found in principle that PMN MDC are usually highly present, higher frequency than monocytic MDC, which can be due to the fact that they have a much quicker PMN MDC, much quicker turnover, and they are dying relatively quickly. That's why the production is higher. This PMN MDC, so they also accumulated strongly with the tumor progression, and they show also immunosuppressive pattern, and as well as monocytic MDC. You see that, for example, polymorphonuclear MDC showed higher expression of CD39 and CD73 than monocytic MDC, but at the end, the immunosuppressive activity of both cell population is more or less the same and very strong. Again, these cells were isolated from the mouse tumors and incubated with activated T cells. Now, as I told before, that it's necessary for these cells to migrate from the bone marrow to the tumor, and they use chemokines and chemokine receptor expressed on these cells. And PMN MDC, for example, are very prominent for the expression of CXCR2, for example, whereas monocytic MDC express mostly CCR2. So it means that there are different chemokines which will be involved in the migration of both population in the tumor microenvironment. With regards to CXCR2, expression. So PMN-MDC really the champions among all or most of the immune cells you see here. So therefore, the ligands, so for CXCR2, like CXCL1 and CXCL5, are particularly important for migration of this subpopulation. And you see that there is a huge also difference gradient in the concentration in CXCL1 between tumor microenvironment and plasma. So it means that there is a kind of pump which can just facilitate the migration of PMN-MDC. Now also what is very important, that the concentration of CXCL1, for example, in the tumor microenvironment is positively correlated with the frequency of PMN-MDC infiltrated tumors. Now, we're told about mouse, but of course, important for us to know what happened in the human settings. And we studied melanoma patients, so with advanced melanoma with metastasis already, stage 4 mostly, and non-metastatic patients, so mostly stage 1 and 2, and found that there is a strong accumulation of PMN-MDC in patients with metastasis in comparison with those without metastasis. Moreover, especially MMDC strongly express PD-L1 in metastatic patients. So again, most important that both PMN and MDC showed just before the start of the therapy a strong inhibition of T cell proliferation. And you see that there is a quite high heterogeneity between the patients. So some of them, MMDC showed very strong inhibition of T cell proliferation. In others, this inhibition is not so strong. And this could have, of course, also impact on the sensitivity and responsiveness to the treatment, which I will show later. Again, the cytokines involved in the activation and generation of MDC are accumulated in the plasma of metastatic patients. So among them, IL-6, IL-8, and TNF-alpha. The also important point is that the high frequency of PMN-MDC or higher frequency PMN-MDC and MMDC in the circulation in the periphery actually correlated with the lower overall survival or progression-free survival of the patients. Now, the response to the treatment. So, of course, now the most important treatment in melanoma is the immune checkpoint inhibitors or antibodies against CTLA-4 or against PD-L1. And you see that the patient treated with ipilimumab, the antibodies against CTLA-4, non-responders showed the increase, despite the treatment, increased in the amount of, or at least not decreased the amount of monocytic MDC. This is also corresponding to the higher concentration of factors involved in the generation and activation of MDC, like IL-6, IL-8, or some tendency also for TNF-alpha. And what is also important, that responders, you remember that we had this very high heterogeneity in the response of the patient to the MDC from patient to this immunosuppressive activity. So responders actually show in the before start the treatment again, show relatively low inhibition of T cell proliferation in comparison to non-responders. Just to sum it up, so we have in chronic inflammation conditions typical for the tumor accumulation of cytokines, chemokines involved in the inflammation, leading to the accumulation of myeloid-derived suppressor cells. The activation could be supported also by the factors produced by activated effector T cells and NK cells. Also important factors like S100, A8, and 9, and HMGB1 errors involved in the generation of MDC, which we will discuss a little bit later. But there is also another factors which involved in the MDC generation is extracellular vesicles. Actually, what is interesting that the previous dogma was that actually myeloid-derived suppressor cells could be only immature cells due to the partial block of differentiation of myeloid cells. But now we know that it's not only, it's still true, of course, but it's not the only source of generation of MDC, because different factors produced by tumors could somehow convert already fully differentiated myeloid cells into immunosuppressive cells. So we start with exosomes. So as you know, these small vesicles are produced by also by, well, in general, rapidly proliferating cells, but also by tumor cells, and they could have all possible proteins, which is typical also for parental cells, or cells from which they produce. And I would want to stress your attention, focus on this heat shock proteins, so S100A and 9, HGG1, and microRNAs, which are typical also accumulated in extracellular vesicles. So, but we started with vesicles first and found that vesicles isolated from mouse melanoma cells so could strongly upregulate PD-L1 expression after only overnight incubation on the surface of CD11b GR1 double positive cells from bone marrow from normal mice. These myeloid cells from normal mice after the treatment are able to inhibit T cell proliferation, so they are becoming immunosuppressive. What could be the mechanism of this? So we found that using toll-like receptor knockout mice, that toll-like receptor 4 especially is involved in this process. You see that myeloid cells from bone marrow isolated from these mice are not able to upregulate PD-L1 after the treatment with tumor exosomes. And actually, what is important again to stress that the inhibition of T cell proliferation was completely blocked if these myeloid cells were isolated from toll-like receptor 4 knockout mice. Which ligand for toll-like receptor could be? So after long studies, we found that red exosomes, so exosomes from red melanoma cells, expressed a high amount of heat shock protein 86, or another name, heat shock protein 90 alpha. And if we knock out this gene coding this protein from melanoma cells and isolated exosomes, so we were not able to see the inhibition of T cell proliferation by myeloid cells after the treatment with these exosomes. So this interaction is then important, leading to the acquisition of immunosuppressive activity. The same we found for human melanoma cells. Again, exosomes can upregulate PD-L1, and this after treatment of monocytes, normal monocytes, and these normal monocytes acquire the immunosuppressive ability. Again, this PD-L1 expression and also PD-L1 expression and is depending on toll-like receptor 4 and depending also on the expression of HSP90 alpha on these melanoma cells, and then also, of course, in exosomes. And of course, after the if we treat monocytes with exosomes isolated from melanoma cells, depleted or knocked down with for HSP86 or HSP90 alpha, so we did not see the inhibition of T cell proliferation. Now, let us then look on which particular components of exosomes could be important for the immunosuppressive activity. Let's focus on heat shock proteins. Of course, heat shock proteins could be both in exosome but also in soluble form. And just to look on the significance of the soluble form, so we use the recombinant HSP90 alpha. We treated CD14 positive monocytes and found again upregulation of PD-L1, which is very much dependent also on the expression of toll-like receptor 4. You see the antibodies can block this upregulation, and also this depends on NF-κB, which is probably the main factor of this PD-L1 upregulation in normal monocytes after the treatment. Moreover, the concentration of HSP90 alpha was depending on the frequency of monocytic MDC in the circulation. Moreover, after the treatment, so these monocytes acquire immunosuppressive activity, as you see here, which is actually completely abrogated by the treatment with toll-like receptor 4 inhibitor Rezatorvit or with antibodies against PD-L1. Now, what else do we have? We have also S100A8 and 9 and HMGB1. Together with HSP90 alpha, they belongs to so-called DAMPs, danger-associated molecular patterns. And actually, with these factors is a little bit more complicated because they have as receptor not only toll-like receptor 4, but also RAGE, receptor for advanced glycation end products. And you see these interactions are quite complicated. Just to decipher the importance of both receptors and both factors like S100A9 and HMGB1, we again treated with them monocytes, and we saw that both factors can induce the immunosuppressive activity of monocytes, although you see already here that HMGB1 is a little bit weaker inducer than S100A9. And again, using the treatment with respective inhibitors like Rezatorvit or FFPSC1, we found that this induction of immunosuppressive activity could be abrogated. Both factors as a mechanism can upregulate PD-L1 activity, and which is actually here I show on list 109, but it happens also with HMGB1, which is actually blocked by toll-like receptor inhibitors or RAGE inhibitors, although you see toll-like receptor inhibitors can work more efficiently. Again, we see the production of reactive oxygen species by monocyte after the treatment, which is also blocked by respective receptor inhibitors. And what's important also that after the treatment with S100A9, you see the induction of IDO, the enzyme involved in the degradation of tryptophan, essential amino acid important for T cell activity. And this induction was completely blocked by the treatment with toll-like receptor 4 inhibitor, but not with RAGE inhibitors. So it seems now we have also some clinical or human data where we show that the concentration of S100A9 in the plasma positively correlated with the increased frequency of monocytic MDC expressing PD-L1. Then after the treatment with immune checkpoint inhibitors, so in the responders, so we saw the degradation or decreased in the amount concentration of S100A9 in the serum, and higher concentration of this factor in the serum or in the plasma actually negatively correlated with the survival of the patient. So And also, again, we can use these factors as factor distinguishing between the responders and non-responders, since responders in the baseline and the time of before treatment, so they show lower concentration of S100, 8, and 9 than non-responders afterwards. What about microRNAs? So this is a small molecules RNA, so having the regulatory capacity involving in expression or inhibition of activity of some genes. So we studied this in mice, making the isolating CD11 BGR1, just normal immature myeloid cells, and making the transfection with microRNA complexes. And we found that there is an upregulation of PD-L1 activity again on these mice, on these myeloid cells from mice. And among these different microRNAs, so we saw that mostly microRNA 125A has a strong upregulation of this PD-L1 activity. We wanted just to decipher which mechanism or which factors could be involved in this, and we make a microRNA analysis of the monocytes, of myeloid cells from bone marrow treated with microRNAs 125A, and found actually the accumulation of some genes which are known for induction of immunosuppressive capacity. And we found that actually on RNA level also there is an upregulation of ions, for example, IL-10 and PD-L1. With IL-6, which we could confirm also on the protein level that there is an accumulation of IL-6 in the supernatant. There is an increase in the reactive oxygen species activity, and when we wanted just to look on the mechanisms, so it was known that toll-like receptors 7 and 8 are important for the interaction with microRNA, and we used knockout mice. Unfortunately, we did not find, to our great surprise, the involvement of these molecules. And the only thing what we know exactly that the involvement of NF-κB, since NF-κB inhibitor can completely block the expression of PD-L1. And we're studying now further which receptor could be involved in the interaction with this microRNA. So now in the second part, we're coming to the inhibition of activity of myeloid, targeting of myeloid-derived suppressor cells. Since there are so many mechanisms, difficult to find some really magic bullet which can target one molecule leading to the inhibition of both PMN and MMDC. At the moment, so we have like three classes of inhibitors. Some are dealing with the inhibition of immunosuppressive activity, others with the regulation of myelobases or MDC depletion, and importantly also dealing with the blockade of MDC migration. At the moment, so there are like in the world like 36 clinical trials in cancer patients, and 6 of them are dealing with the combination with immune checkpoint inhibitors. So I will shortly show you that examples from all 3 groups which we studied. So first is using sildenafil or Viagra is an inhibitor of phosphodiesterase 5, which leads also to the in MDC to inhibition of nitric oxide production through the inhibition of iNOS activity. When we treated with sildenafil, the mice having already tumors, this in this spontaneous mouse model, so we showed a quite impressive anti-tumor effect, which is correlated with the strong decrease in the production of nitric oxide, which is expected by MDC isolated from the tumor, but also with a decrease in the expression of arginase 1. And most importantly, that sildenafil-treated mice, they are MDC isolated from the tumor, so they're less immunosuppressive than in the control mice. Then we started together with Jessica Hassan from National Cancer Center in Heidelberg, a small clinical trial where we treated patients, actually melanoma patients, which received, as you see, all possible therapies before, and were completely, at this time point, were completely resistant to all these therapies. So we treated them with escalated doses of tadalafil, which is actually long-lasting inhibitor of phosphodiesterase 5. So then you don't need to give this medicament every 3-4 hours, but it's enough to give once per day. And we found actually that in 3 patients, so out of these 10 patients, so we see the at least stable disease or partial response. Now we've looked on the situation in the peripheral blood, and we found that after treatment in stable disease patients, so we found a decrease in monocytic MDCs, which is not found in the progressive disease patient, and also in the tumor, because we took also tumor from the skin tumor biopsies. We found also decrease in the production of nitric oxide in tumor associated monocytic MDC in patient with a stable disease. And in turn, there is an accumulation of CD8 T cells, and especially what is important, the increase in zeta chain expression, which is important for T cell activity after the treatment. Now, the second example I want to show you is dealing with the migration, blockade of the migration, because the main events are happened in the tumor microenvironment. Therefore, it is very important also to somehow to block the entrance of MDC in the tumor. And one possibility is just to use some inhibitors of CCR5. CCR5 is a chemokine receptors expressing different cells, but what importantly expressed very strongly on MDC. And we found that actually MDC expressing CCR5, they show higher immunosuppressive activity than MDC negative to CCR5. That's why it makes sense probably to block the emigration in the tumor. And really, while using this fusion protein immunoglobulin against mouse CCR5, we found that treatment of tumor-bearing mice in Ren transgenic model leads to the increased survival, and some of mice, they even survive for a long term and even don't have tumor in the end. What is important also, of course, to know how this treatment would somehow influence the migration of other immune cells, and especially important was to see what happened with T cells. So migration of conventional CD4 T cells and CD8 T cells was not damaged, whereas the migration of CD4 positive Tregs was downregulated, which is also good, like additional effect, not only migration of MDCs, but also Tregs. And also the MDC after the treatment, they express less nitric oxide. And then, of course, you see the decrease in the amount of MDC in the tumor of treated mice. We are trying now to make a clinical trial in the clinical settings, because there is, and as I showed you here, probably, this one, Maraviroc, is the medicament which used for treatment of HIV patient, which is already approved for this treatment. But this is actually the also inhibitor antagonist of CCR5, which can use also, of course, in the clinical settings. And now, just coming to the conclusion. So MDC are generated, activated, and enriched under chronic inflammation and tumor progression. There are two ways of MDC generation. So one is impairment of normal myeloid cell differentiation, and another way is conversion of normal mature myeloid cells, in particular monocytes, into MDC. And this is really an important question. So in the clinical settings, which way is more prominent in one or other patient? And therefore, one can use also different strategies to target these myeloid cells. So MDC can strongly suppress anti-tumor activity of T and NK cells via multiple mechanisms. So enrichment of circulating MDC correlated with worse outcome in cancer patient, and MDC targeting could be used as a powerful tool of combinatorial tumor immunotherapy. It would be important, particularly to use this in combination with already existing immune checkpoint inhibitors. And in case we would need just to activate T cells in case of so-called cold tumors, so one can use the vaccination or adoptive T cell transfer, which can then be combined with MDC targeting. And I would like to acknowledge the people from the groups who worked in this project, and also the different collaborators from different countries, Israel, USA, and Italy and Germany. And just to show you two places where we are working, at German Cancer Research Center here in Heidelberg, and this is University Medical Center in Mannheim, where we have the very important center for treatment of melanoma patients, so where we can get also our clinical material. So, and thank you very much for your attention.